I think people work all their lives for an opportunity to discover and develop a drug like crizotinib.
It's a very touching and exciting moment for me.
We have the opportunity to make a difference for patients.
This is probably one of the most significant developments in lung cancer in a long time.
It is changing how we are viewing the drug development process.
I think the crizotinib team is, their dedication is unequaled.
I just have a big smile on my face every day knowing what we're able to do for patients.
This is a breakthrough.
Over the last 10 years, we've learned a lot about lung cancers and how there are actually different subtypes of lung cancer defined by different underlying genetic mutations.
It started from CMAT project in the year 2002, and we have the big breakthrough.
Around 2005, we identified a molecule called PF2341066 that was indeed a potent and selective inhibitor of MET. We continue to fully characterize this molecule and identified that there's another target besides CMET called anaplastic lymphoma kinase, or ALK.
We thought it was going to be just your typical phase 1 trial.
When crizotinib first entered clinical trials, it was an interesting compound against a very intriguing target. I think what happened subsequent to that, no one could have imagined.
To raise another highly effective drug, we have to find out the real target in every cancer. So when I found EMF4LK, I instantly realized that will change both of diagnosis as well as therapy for lung cancer carrying this fusion gene.
In 2007, there was a paper published in Nature that described EMF4-ALK as a fusion protein. Jamie Christensen from the Discovery Group emailed me the article and asked would this be of interest to us for our clinical trial. I read the paper and said yes, of course, because we knew this compound was effective against ALK. We said, really, we need to change our way of thinking. Let's not just look at CMET, let's concentrate on ALK.
We really adapted very quickly to this information to amend the protocol and start to focus on ALK-positive patients.
The first patient we identified with an ALK rearrangement, we started him on, I believe it was December 26th of 2007, which is interesting because that's about 4 months from when Dr. Mano first reported the discovery of ALK.
The first time we heard about crizotinib, we got to hear about patient tumors were responding, and it was really kind of almost a get-up-out-of-your-seat moment.
The reaction of the medical community was quite positive.
They realize that Pfizer's really serious about personalized medicine.
This was an important new treatment, and that they would like to see and review that data.
The way a company is supposed to develop a companion diagnostics, all the coordination from the beginning to the end are exactly what we did with Xalkori.
The crizotinib team has just worked around the clock to get not only the submission in, but then also the approval, dealing with all the queries from the FDA.
That's really exciting, to see Xalkori get approved, and this is a dream come true for medicinal chemists.
My dream has come true.